today is Bumi Abel. She's a computer scientist who's been with the clinical neuroscience department since July 2008 in the laboratory of ALSoD. She received a bachelor's degree in computer science and a master's degree in computer systems and networking, and now she's in her fourth year of a part-time PhD in bioinformatics. She currently works on amyotrophic lateral sclerosis on the online genetic database, which is ALSoD, which is basically an open source data repository that provides both the scientific community and the wider public with up-to-date information on ALS genetic research.
Thank you, Ashley. Good afternoon. I'm here to speak to you on the ALS online database, ALSoD. This has been going on since 1999, but I took over 2008. My name is Bumi Abel. Because of the mixed audience, I'd like to start with defining what motor neuron disease is. And it is a progressive incurable disease, neurodegenerative disease that attacks the motor neurons. Patients have symptoms like weakness and wasting of muscles, loss of speech, swallowing problems, and breathing problems. There are 4 types of MND, and they are ALS, which occurs in 60 to 70% of cases, PVP, which is progressive Volcker palsy, 20% of cases, progressive muscular atrophy, 10% of cases, and the primary lateral sclerosis, which is very rare, but eventually ends up in ALS patients. The first publication to talk about the amyotrophic lateral sclerosis was by the French neurobiologist Charcot in 1869, and the first celebrity who had the ALS online database, ALS disease, was Lou Gehrig, and that's why in America the disease is known as Lou Gehrig disease. So he was the first person to have that and brought to light that disease. And amyotrophic means without nourishment to muscles. That is where the loss of signals to nerve cells from nerve cells to muscle cells are hindered. And there's a lateral means to the side, which is the location of the spinal cord. Sclerosis means hardened nature of the spinal cord in advanced cases. ALS is also known as Charcot disease, like I said, or Lou Gehrig disease, depending on where you're, what part of the world you are. And it's a complex neurodegenerative disease, killing patients within 2 to 5 years after diagnosis. And most of them die from respiratory failure. One of the people living with ALS at the moment, which we may all know, is Professor Stephen Hawking. Is currently living with ALS, and you have, like I said, weakness of muscles on the limbs. And from that diagram, you can see there that a normal muscle will have that with the nerve cells intact and everything, but the affected muscles, when they begin to waste, becomes very lean and causes all sorts of complications for patients. Now, about 10% of cases of ALS cases are familial, meaning there is 1 or 2 or more people in the family that has got that disease, but majority of the cases are sporadic, like that SARS, and this is, probably due to a combination of genetic and environmental factors. And studies have shown to us that people in the military, people who do sports are more prone to having this disease. They've got higher risk factors. And now coming to the genetics of ALS. On ALSoD, when I took the statistics of the gene discovery in ALS in 2012, was about 2 years ago, found that genes keep keep coming up every time there's a GWAS or a large study, and researchers keep finding many genes are associated with ALS every now and then, and it keeps increasing. It wouldn't go down, it keeps increasing. So like I said, now it's more than 96, but that was done in 2012. So due to next generation sequencing technology, it enables more data to be produced by researchers when trying to find genes that are responsible for ALS. And due to the large volume of data produced every now and then by researchers, it's important for these genetic data, to be put in like a repository so they can keep tab on all of this data. And this brought us to the ALS Online Genetics Database, ALSoD, which was originally designed in 1999 for sharing clinical and genetic data. It's publicly available. You don't need to log on or have a username and password to go on the database. However, for you to submit mutation data, you need to log on, because we want to show the authenticity of people who submit mutation data on the database. And it's useful for researchers and non-researchers. It links to other genetic information sources. So it's left the stage of just being a storage facility. That's why in the older version, it was just ALS Online Database was just to deal with the SOD1 mutation, and that was by sorry if I pronounce it wrongly, Radinovic. In 1999, this was when the database was created, and it was just for gathering gene mutations of, mutation data of ALSoD1 gene. And it was then the Amyotrophic Lateral Sclerosis ALSoD1 which brought the name. It was on the old URL ALSoD.org, and as time went on, 4 genes were added, and from where I took off from, there were just 4 genes on the database. But the newer version since 2008 is known as the Amyotrophic Lateral Sclerosis Online Genetics Database with a small O, not just for ALSoD1 anymore. And it's on that URL, also.iop.kcl.ac.uk. It's got 113 ALS-related genes at the moment. It's gone from being a storage facility into analytical tool, collation and integration of genetic data. And it's got graphical representation of all this data. An overview of the gene page is for every gene, for all the 113 genes we have, we've got this template for the genes. There's a brief information of that gene. We have the graphs and statistics, which shows the bulbar and limb ratio, files/sols for every gene, and it links to external information, like we have there, the bioinformatics section, where you can click on it and it takes you exactly to that the web page talking about that particular gene. One of the recent additions to the database is the animal models, where you have publications from animal models just for people who do research on animals to see what animals have been researched on that particular gene. And we've got the key publications right at the bottom, the long list of it. We've got the detailed analysis of patient data and mutation data we have on ALSoD. And once you click on any gene or more than one gene, click some criteria like the age of onset, is it SARS or FALS, or the gender you want. It shows you the gender ratio, the family ratio, the site of onset ratio, and even the countries where all these mutations have been found. Gene interactions are predicted using the GeneMania bioinformatics tool, and this is a collaboration with the GeneMania team, where you can select the genes you want, and it displays the graphical view there. Like you can see, it shows how the genes interact, number of genes you select, how they interact with each other and one another. Comparing genes on the mutation and patient data in ALSoD, you can just click on, that's another page, you click on any two genes you want to compare, like the FOSO and the TDP43. It shows you the statistical summary and the countries where these genes are found. Click on that. Also, it predicts pathogenicity of mutations, where you can click on a gene, click on the mutation you want, and it shows you if it's pathogenic or not. And this was actually done with 3 bioinformatic tools: the Panther, the SIP, and the Polyfen. So when you have, when the system goes through those 3 bioinformatic tools, it tells you if they're all saying the same thing or they're saying something different, and the conclusion of the all matter if it's pathogenic or not. And it also shows you the replicator studies that have been done for that particular mutation. Also, you can view charts as well. And another analytical tool on the ALSoD is gene credibility. Here, it helps those who are coming into the field of ALS maybe for the first time, or who are actually interested in knowing what genes are actually more important in the field of ALS, or what genes do you think I should be looking at? And it helps to give people direction, researcher direction on how to go about their research. And so when you click on the criteria on the database on that page, you can click on controls, ranking the genes by controls or by codon, by the mutations, or by the number of cases that have been researched, or the number of publications. Once you click on whatever you want here, I've clicked on everything, and the patients and mutations are compulsory criteria in every selection. So once you click on that, it ranks the genes, and it tells you, okay, with all of this criteria you've chosen, ALSoD1 comes up first, then the FUS, and the TDP43. So this is actually dynamic in the sense that once we have more data on a particular gene, let's say like the DAO, if we got more data on the DAO, like so many people are making research on that, and they keep updating the database with their data, it may go from that rank, from that very bottom to wherever, but of course ALSoD1 still remains there. And so we encourage researchers to submit the data every time they find something. And the ALS Online database also displays genome-wide data. Initially it was 5 populations from Professor Amar's team, and then it developed into 8 populations. And at the moment we have a recent publication by Dr. Fogg, where there's a genome association meta-analysis identifying a novel locus at chromosome 17. And this was done just late last year, and it's all there where you can just, if you find a SNP in your research, just type on, query the database by typing the SNP ID, and it tells you what the p-value and some other information you might want to know. So the other thing about the database is that it's mapped to, the gene variants are mapped to genomic coordinates, where you can see a summary of the mutations, and it tells you the HGVS codes, so you could refer to it, the SNP IDs, and you can refer to it in all the databases like in the portal SwissPod. Also with the help of some of external bioinformatics people, we were able to find more of the HGVS codes using the 1,000 genome and the exome variant sequencing database. So population frequency of every mutation in ALSoD is displayed on there, on the database. And the animal models, you have the publications, you have a summary of the names, you have all these links you to OMIM, links you to KEGG for that particular gene, and you have all the publications stated there. Another thing we did on the ALSoD database is, or what we realized is, so many people use the mobile devices like the phones and tablets to search for information, and it could be too small when you use your phone. If you have to check the full version, it could be very tiny. It's a genetic database, so it's got loads of information. But we had to do a summarized version of the database by creating a mobile platform where you could, you have a summary of the full version. It's also, and there's also an app for the Android market. That's the Play Store. It's a switchable view where you can, from your tablet or from your computer, you can switch. You can click there to switch to a desktop view on your mobile, or on your desktop switch to a mobile view. So it's a mobile website and it's an app, and there's increased access to the data we have on the database, because through our analytic monitoring system, we found that more people are using the mobile devices to get the information. They want to be on the train, or you're just in a lecture room like this, you want to check a particular mutation. You say, oh, I found this mutation in my lab. This is, how many people have it, what countries they have it in, and has anybody found anything, and that's that gene. So you can use it anywhere, as long as you have your internet connection. And then this helps to encourage collaboration with other teams. You may want to ask me, how popular is this database? Is it just used by IOP students, or is it just used by clinical neuroscience, or marketing only? But I'll tell you, it's used widely worldwide from different countries, and we've recorded increase in the use of ALSoD year in, year out, and it's updated regularly, and we have the largest number of people from the United States. So this unknown is, it has to do with IP addresses and all that, but you have more people from the United States, from the UK, accessing the database. So this is a typical day, like yesterday. I had to do this before coming in here to find out what those who accessed the database just yesterday. We had some 59 visitors. The future of ALSoD. We're hoping to collaborate with Patients Like Me, which is a database where patients data, or it's a patient community database dealing with more only patients, and I think they talk to themselves on share experiences. So we're hoping to collaborate with Patients Like Me in the future. We're hoping to integrate with external databases, and we're hoping that mobile applications on a wider platform like the iOS will be developed soon. I'm not sure about BlackBerry anymore because of, where it is. But I was thinking I would do the BlackBerry before the iPhone, but it's okay. And then we've got the structural display of variants, which we're going to be collaborating with other teams externally. So I'd like to appreciate the people who gave the original idea of ALSoD, my current coordinators, Amar's team, and external help from Simon Talk and Juan Lin. They've been very, very helpful. Also to the funders and to the sponsors. Thank you very much for listening to me.
Thank you. It's very, very useful for business.
That's where the database sits. Is it on a server here?
Yes, it was on ALSoD.org, but when there was problem with domain name of people not, fundings and all that problem, it was shifted down to the VM3 server at the Institute of Psychiatry. So it's on iop.kcl.ac.uk.
And where does that service sit?
It's
Building, is it?
KCL, not, I don't know where it is, but it's KCL controlled. So if I have a problem, I know who to call.
Ian.
I don't know if the server is actually on the IOP or it's somewhere in Strand or what to do. I know it's VM3 server. That's all I can tell you.
Okay, thank you.
I have a question. Was it criteria of genes making it onto the database? Do they have to be replicated? Do they have to get persistent samples on?
Of genes on the database. Genes are actually submitted on the database once they're published. So most times I have to go through the papers and, or maybe one of the team will tell me, oh, there's a gene available now, has been found in ALS, or maybe the gene-wide study, or even one of the researchers will say, oh, I have a gene just been published, but it has to be published. But mutations, sometimes you have unpublished ones and published ones, and the unpublished ones, it has to go through the team and the ALSoD team to find out if this is actually authentic, but the genes definitely will have to be published.